Okay, wonderful. So today I'm going to share with you one line of projects that in our lab that started about 2 years ago. So it's on the enhancing protein-protein interaction prediction with deep learning. I think with this group of audience, probably doesn't need much introduction why protein-protein interaction is important. So the objective of this project is basically if we have a pair of proteins, we want to know whether these two proteins under some circumstance that will interact with each other. For example, whether they will bind to each other, right? So in terms of the specific task for prediction, then I can see there's at least 3 different variants. The basic, the most straightforward one is to have a binary prediction, whether or not they interact or not. A little bit more advanced, it would be like, okay, what type of interaction it will have. And the in our case, the most advanced, actually, the hardest task is say, well, can we predict the binding affinity as a continuous variable, right? Instead of just categories or a binary indicator. So, and I'm going to be fast here so that it's actually, if we can predict a protein-protein interaction, it will help us to understand like cellular biological function, and we can also actually use it to enhance our knowledge of the biochemical cascade of the disease, and to also facilitate the drug design. So there has been quite a few different approaches being proposed for protein-protein interaction prediction. We grouped them mainly into 2 categories. The first category is basically using traditional statistical learning approach. For example, K-nearest neighbor, support vector machine, or some statistical test. The limitation of this group of approach basically, they mainly rely on quite sophisticated feature engineering, and using the statistical model built on top of that. So, which means that if the feature that used as input to the model, right, somehow have limited coverage of all the important aspects towards protein-protein interaction, then the model itself would carry that limitation. The second group of approaches is actually newer approaches, but is deep learning approach. So for deep learning approach, so far it has been like mostly a convolutional neural network based approach. For those approaches, we know CNN actually is not designed for capture sequential features. So basically for this type of approach, they pretty much lost the contextual and sequential features of the protein sequence and also between the interacting pairs. And some of the approach, such as DPPI, right, require quite extensive pre-processing because they have to use SciBlast to pre-process all the known sequences and then to build a map of possible similarities. So in our approach, we call it PIPR. So it's a deep learning approach. It has several major, this is overall diagram, and it has several major steps. So the first step, at the very bottom here, we start with the protein sequence that we care about. And then we want to derive feature space with what we call embedding. So it's a high dimensional vector space, typically 200 to 300 dimensions. And for each individual amino acid within the sequence. And then the next step is to use RCNN, a recurrent convolutional neural network, for each of the protein sequence to learn a sequence embedding. So basically one vector to describe each protein sequence. Since we're concerned about the PPI, so we are going to have this element-wise multiplication between the 2 sequence vectors there, and to generate another vector to describe the interaction. And then it's followed by the multi-layer perception, and then followed by this the individual prediction task. Here, I mentioned that we have 3 different tasks. So that's the main idea. And then what I want to point out is that this RCNN actually the same model for different the protein sequences. They have shared model parameters. Okay, and so I'm going to show you. I mean, this is a very straightforward idea, right? So I'm going to show you how it works. So the first task is the binary prediction of whether or not there's an interaction between two proteins, and we compare with a group of as a deep learning based approach. And our PIPR, you can see that in terms of accuracy, F1 score, and MCC, so they all achieve a better result over the previous approach. It's I think it's quite impressive here. So it's all 90-some%, right? So you can see that for different previous different deep learning approach, basically they use a lot of convolutional neural network, so that they lost the sequential information. So so that's the binary classification. So now we move to the comparing to the more traditional approach. So traditional statistical learning approach, I mentioned before that they require sophisticated feature engineering, and you can see that several of these approaches use features of autocovariance of the physiochemical properties of the amino acids and the proteins. And some others use actually the 3 descriptors, compositions, and transitions, and these distributions. And those are the feature space that are being typically used. And then there's other variants of that. So that you can see by comparison of this, again, this paper is achieved quite impressive performance over this traditional statistical learning approach. So in terms of the multi-class prediction, here we want to predict the interaction type. There's 7 different types I listed here. And you can see that, again, we compare with a group of different approaches, those baselines, and basically either majority or random, and also statistical learning approach, and also deep learning approach. And we group them into the 3 big categories. And you can see that we have the highest accuracy here over all the approaches, and actually it beats the second best approach. It's right here, so it's quite a lot. Of course, this average, because it's multi-class prediction, is not as high as this 97% from the binary prediction. The last task we tried is to predict the binding affinity, and the binding affinity. Here, we again compare with both the statistical learning approach as well as the deep learning approach. So we use the MSE as MAE as and also the correlation as a measure. You can see for MSE and MAE, the numbers, the lower the better, right? Because that's measures the error. And as for the correlation, of course, the higher the better. You can see again that we achieve the lowest. Let's take MSE as an example. We achieve the lowest MSE, and then all this previous approach. Okay, we beat them by quite a wide margin here. And also we have this highest correlation. And the second best would be this other actually machine learning, deep learning approach based on the creating the embedding of the protein sequence. Those 2, right? So, but still we beat them in quite big. I'm showing you actually a specific example here that we see, because we did look into the different examples to see that whether our prediction can bring any biological insight. Right? So in this particular example, you can see there's this blue protein and the red protein, right? So I zoom in the little here, and then the yellow dot here is actually there's a mutation on the chain of the red one, right? So it's highlighted with the this yellow dot here. So, and then there's some mutation. This on the left side is the wild type, and on the right side, this is the mutant type. And because of the mutation, actually, the red one actually has some conformational change. And because of that conformational change, you can see that the binding affinity also changes as a result. So in this case, you can see I showed that on the table here, the ground truth of the binding affinity and our prediction. Okay, you can see we're getting quite close, even though not exactly what the ground truth is, but actually in the ballparks there already. So the take-home message here is that the PIPR is actually end-to-end framework, and also it's what all we need is that you have the input of your protein sequence, and then we can fed into this model, and then we can use this to make a quite accurate prediction. And what I didn't have time to show you here is actually the running time, right? So ours is ultra much faster than most of these other approaches. And because all the other approaches need to process, especially deep learning approach, they need to process not only the set of proteins that you care about, but all the other set of proteins that could be, possibly used maybe from different species. So they have to process a huge database for this. But in our case, we only need to process maybe the set of proteins that you care about in order to get quite a good result. But of course, if we have the capacity to examine more proteins, then the result can be further improved. So for the last few minutes, I want to share with you a more recent result that we have. So we want to apply this to the COVID and the SARS-CoV-2 virus, right? So the SARS-CoV-2 virus also has its own protein, right? The virus protein. We want to see that whether we can predict the interaction between the human protein and the virus protein. And there has been last year a nice work published on that, right? But they're not doing computational approaches. They're actually the experimental approach to measure whether there is interaction between a given virus protein and the human protein. On the chart I showed here is that actually the purple ones in the middle here, those are the virus protein, and the orange ones on the circle, those are the human proteins. That's in that nice, I think it's a Nature paper, and they already identified quite a lot of those interactions, right? A few hundred of those. And on the right-hand side, I'm showing that those are the virus proteins, and there has already some limited number of Go annotations associated with that. Okay, of course, you can think this both data set actually are not, we can think of that they're not complete data set because the experiment are still going on, right? Because the SARS-CoV-2 is relatively new. That's why we hope that see what if we can do to predict some possible interactions. And so, of course, I want to mention that this is not limited to what I just showed you on the last slides. We hope to be able to integrate as many as other knowledge base into this, because the actual sequence data, interaction data is very limited in that regard. So we want to be able to leverage other existing knowledge that we already acquired. For example, from the drug bank about potential drug target, and for example, from the strain database, and about the functional proteomic analysis. So we want to be able to integrate other knowledge, the prediction of the interaction. So here are some of the knowledge base that we have used so far. And then our idea is still like what I just said, that in a PIPR project, that we want to map them to create semantic embedding space to describe the data, right? To describe the information either in the data or the knowledge base. And also we want to integrate it between across different embedding space so that it can help us to do the prediction. So I think I'm pretty much talk about this slide. So we want to be able to integrate across different, either the data space and the knowledge space across different space. And the idea of doing this is that actually we want to have a two types of models. One type of model we call the knowledge model. So basically within each space, we want to create an embedding to describe the data in the space, for example, the protein sequences in the PIPR project, or the gene ontology, right? So those are individual spaces that we want to model using the embedding. And also the second type of is, here is called the transfer model. So basically we want to be able to link the information, the data, the knowledge across different spaces. We want to be able to transfer knowledge from one space to another, and so that we can do an integrated prediction. Therefore, our approach is called JOIE, and it's really joint embedded learning. So again, those have two components. One is that, okay, we have the knowledge model for each individual domain. And then there's quite a few knowledge graph embedding approach that we could use. And I just list the three here, like Transy, Dismount, and JOIE. And those are some representatives. There's even newer ones come out. So you can use any of those to do that. And and then the next would be the transfer model. So basically those are the defined between pairs of the domains, right? Here I'm showing you that between the proteins and also between the proteins and the gene ontology. So you can see those are links here, and actually those help us to link between different spaces. And we use this transfer model to map that. The idea here is that because there are two individual embedding spaces, so we want to be able to create a mapping that project everything from one space, let's see the protein space, to the gene ontology space, such that if we know that because of gene ontology annotation, right, so one protein have certain function, or here is a subcellular location, and then we expect those in that, once we map to that same space, and then they are very close to each other. So that's the idea. So, and again, the overall loss function basically is the sum of the individual loss function within each individual space, and together with the loss of the transfer model. So here is the what we did in the experiment. So we want to do the prediction of the interaction between a human protein and a virus protein. And we take the data from the paper I mentioned. And then so because this was published more than a year ago, so since then there's some new data come out, then we just use it as validation. So here I'm just showing you the F1 score, and as a function of the what percentage of the data we use training versus testing. And so you can see that, of course, the more training data we use, right, so the better the result. And also here the different red, blue, and green line shows that what's the how much more information we incorporate in that. So there's no human PPI, there's no like human and the SARS-CoV-2 PPI, there's no interaction between SARS a few years ago, between human and SARS, and also MERS, so that after we add one more domain in it, actually you can see the result gets better. Here I just want to show that in this set, it's a specific example, it's a human, it's a virus protein, and those are the top predicted human target protein, and the green ones is already being validated. I mean, it's already in the paper, so they already published that. The yellow ones actually is what we predict as highly likely, but then later get verified, and then the blue ones that are yet to be verified. So you can see that it's a very actually I think it's quite a interesting result. So we can indeed making some good predictions that to maybe provide scientists with some candidate for hypothesis test. So I'm running out of time, and I would like to leave a couple of minutes for questions at the end. So, but before that, I really like to thank all my students, postdoc, and the collaborators here. Thank you.
Thank you so much for the wonderful talk, as always, and for keeping it within the time. I actually do have a lot of questions, but before I ask, let me just check if there are questions from the audience. Okay, there's no, that's great. So let me ask some questions. The first one is, what is the advantage of using this RCNN over using something like protein BERT, some pre-trained models?
So for the I think for the BERT, well, you can use BERT, but the BERT is trained on the all the protein sequences, right? Unless you train your own, right? So it's on the all the protein sequences. Actually, the so that's sometimes it's if you just care about one species, right? And then the BERT may not give you the best prediction, at least in our experiments, because there's many similar proteins across different sequences.
I see.
So sometimes it's like if you don't have the specific species, unless you modify the BERT, right? So that you start with the BERT, pre-trained BERT, and then you improve upon that. Then you would be able to capture that the relationship between species. What we are showing that is a really, in our approach, is a very lightweight approach. You can start from scratch, right? And then, but it still can get you very good result. It's much faster than any of these other approaches. I forgot to put in our paper, we have I think also on the product website. So has a comparison. It's like ours is like I forgot, a few minutes, and all these others is like many hours or days.
I see.
Sorry. I see. Airport dropped. Okay, so that's why it's very lightweighted, and also it's specifically good. Like if you don't have, like for example, if you have like SARS-CoV-2, right? Before that, we don't have a lot of information knowledge about that, right? So therefore, you can train a very lightweighted model to start to do prediction.
Yeah, that sounds right. So the second question is, with the very recent availability of AlphaFold 2 and Rosetta Fold, which means we most likely will have some reasonable structures to start with, how do you think such structure information will change the state of the art on this problem?
I think that's an excellent question. So one of actually I didn't have time to show is ongoing project is really using the instead of right now I've only shown using the protein sequence, but right now our current approach is actually just using the structure.
I see.
Because previously it's still like only a small subset of the sequences we have the structure information, right? So that's why we have been using the sequence. But our current project is really we directly doing on the structure, and when the structure is available. But we haven't published that work yet. It's still ongoing. We're working on that.
Yeah, I look forward to seeing the results. If I may ask one more question, do you think the same approach can be applied to the antibody-antigen interaction instead of just regular protein-protein interactions?
So actually, it can definitely be applied. I only show the protein-protein interaction as an example. Actually, the same machine learning model I have applied to many different problems. Another, I mean, completely out of this scope. So we have been collaborating with Amazon to use this to predict the product, because you can see the user review as a sequence.
I see.
So it also worked. so definitely you can use this because it's a very general model.
Okay. There's one question in Q&A. It's a follow-up question. So let me ask this one last question. Could you please elaborate further why your running time is much faster than conventional approaches?
Conventional approaches, I want to say conventional deep learning approaches, right? So if you just already have a pre-engineered feature and then you just build a support vector machine, right? So, or some kind of logistic regression, maybe you're faster, but the result is, very mixed. So, but if you just use other deep learning model, they typically need to build a model on the entire protein sequences in order to get satisfactory result. Entire means like not just human analysis, it's like everything there, right? So it's much, much larger than the pool of protein that you may care about. So in our case, actually, we can get from build a model from a small set of proteins, can already be able to extract sufficient information so that can make the prediction already is much better than the previous model, because they typically using the convolutional neural network, which is not the best to capture sequential information, also contextual information.